good. Terrific. So, first of all, I want to thank Professor Komatsu and Professor Ozawa for organizing such a fascinating and unique meeting. I've never been to anything like this, and it's a great honor to be here. I'm going to talk about the neural basis for our very vivid and accurate sense of 3D shape, which is based on image information that we get from surfaces. The experiments that I'll talk about involve awake fixating monkeys viewing three-dimensional visual stimuli while we do microelectrode recording of spike activity in the ventral pathway. Maybe I'll use this, which Dr. Komatsu has already introduced, and the experiments I'll tell you about are in anterior parts of inferior temporal cortex, shown here. It includes the superior temporal sulcus, the floor, and the lateral convexity of the inferior temporal sulcus, and then the basal surface as well. So the hard thing about doing ventral pathway research, many people here know this, is that the domain of possible stimuli is virtually infinite. So it's not like studying MT, where you can define a couple of dimensions like motion direction and motion speed, and then sample those densely, and that allows you to fit tuning functions that basically describe the information that's represented by a neuron. You'd like to do that in the ventral pathway as well, but you can't densely sample throughout all of space, and what people usually wind up doing is random sampling, and that's what's represented here. So we've got some very high and unknown dimensionality. Somewhere in that dimensionality is the tuning region for a cell that we would like to understand. We'd like to know what the tuning dimensions are, what the shape of this tuning function is. And in a standard experiment, random samples in this space, if you're lucky, are going to hit somewhere in here, but just having one or two samples in this region is not going to tell you what kind of information is being encoded about those samples. So a trick that we do in recent experiments is to start out with random samples like this, random abstract 3D shapes, but then create a second generation of stimuli using a genetic algorithm in which higher response stimuli from the earlier generation are more likely to produce partially morphed or conjugated descendant stimuli, which hopefully represent nearby jumps in this unknown high-dimensional space that provide more samples in this region. And then we iterate that across multiple generations with the hope of sampling the peaks, the shoulders, the boundaries of this entire area, and thereby constraining an actual parametric model of shape information that's being presented, that's being encoded, extracted by neurons. The first application of this technique was by Yukako Yamane, and I'll tell you about her experiment on three-dimensional object shape, mainly recording from superior temporal sulcus ventral bank, as Dr. Komatsu mentioned. So this is what the experiment looks like. So Yukako created a custom software package for generating random 3D shapes. The three-dimensionality of these shapes was conveyed by the shading cues that you can see here, and also by binocular disparity cues, which you can't see. So these are all the shapes in the first generation. The average response across 5 presentations and averaged across 750 milliseconds of presentation time for each of these shapes is indicated by the background color, where black corresponds to no response at all, and bright red corresponds to around 40 spikes per second. So first generation, you see that there are a few medium to high response stimuli. In the second generation, you can see that these have produced many different shaped progeny, and the goal of this technique is not to find a single shape, that defines what a cell does. In fact, we've never found that to be the case, that there's a single shape that is a highest response shape, but rather to sample widely in the high, middle, and low response range of the cell, because all of those points are important for being able to constrain a model. So generation 3, generation 4, generations 5, 6, 7, and 8, similar looking sampling. And so now collecting highest response stimuli across all generations, you get this set here. You can perhaps see by eye that high response stimuli vary a great deal in global shape, but they do share partial structure, which is a major prediction of theories of representation by parts, or configural representation, famously expressed by David Marr and Irving Biederman, but actually going all the way back to Selfridge in the 50s. So I won't go through how we parameterize shapes and how we fit models because I don't have much time, but this is a graphical representation of a model, and the meaning of that model can be understood by looking at the projection with tinted colors onto the surface of a high response stimulus, seen from the front, seen from the top. What this model found was that for this stimulus, the region of the stimulus that corresponds to one of the tuning ranges that defines this neuron's response is this horizontal ridge near the front of the object, defined by its three-dimensional relative position, by its surface orientation, and by its surface curvature. So those are the actual parameters of this model up here. And then this cell was sensitive to the combination of a ridge like that and a very low curvature dorsal surface. So how did this kind of shape tuning depend on surface information? Well, what we did after the main experiment was to pick a high response stimulus, a medium response stimulus, and a low response stimulus, and then test those stimuli under different conditions. And those conditions included various combinations of depth cubes, including cases where there were stereo disparity cues, cases where there were no stereo disparity cues, combined with shading cues or texture cues. Texture was not part of the main experiment, but this is an example of one of our stimuli rendered with the kind of textured surface that we used, or nothing else. So the first thing to notice is that if you take away all depth cues, that's the light blue bar here, responses really go away. And this exemplifies what I'll show you later, that neurons really tended to be sensitive to three-dimensional shape, not two-dimensional shape. And if you take away cues for depth, then all cues for depth, you wind up with basically a white silhouette stimulus, and those did not drive the cell well. So these cells were not being driven by the two-dimensional occlusion boundary of these shapes. Texture alone was a very poor stimulus. Stereo was very important. So we got the best responses with both stereo and shading. If you just, and that's the red here, stereo and texture does quite well, probably because this adds, the texture adds a lot of extra stereo information. Stereo alone still elicits very high responses, and shading alone elicits fairly high responses. So in general, what we found was that both shading cues and stereo cues were important for three-dimensional responses in STSV. One remarkable thing was the robustness of this kind of tuning for 3D shape across big changes in shading pattern due to lighting direction. So in our main test, the implicit lighting direction was from the viewer's direction, but you could light from above, from below, from the left, from the right. So we, in control tests, we did a series of below to above, we did a series of left to right, and that's represented by the red and green curves here. And what this shows is that even across these very different images, responses are quite consistent and robust, showing that this is a really high-level inference from shading information rather than sensitivity to a particular two-dimensional shading pattern. So Yukako studied about 300 neurons, and about 100 of them she was able to fit a validated parametric model of shape tuning. And these are just some examples to show, to demonstrate sort of the variety of sensitivity to three-dimensional surface fragments that she found. And in each of these examples, there are 2 high-response stimuli from one lineage and 2 high-response stimuli from a completely separate lineage. We usually, in these experiments, try to run 2 separate lineages so that we can cross-validate and prove that the genetic algorithm is converging on the same solution. And in each case, the model is projected onto the surface of the stimuli to show what was driving that cell. This represents for the 57 cells she was able to run the control test on, the sensitivity specifically to 3D shapes. So this is basically an index of modulation from high response stimuli to low response stimuli with depth cues, and this is the same thing without depth cues, that is the white silhouette stimulus. And most of the neurons are clustered in this region, which means that they were very sensitive to 3D shape, but if you took away depth cues, the 2D shape really didn't drive much of anything. Up here is where you would expect neurons that are in fact sensitive to the 2D occlusion boundary. And there are a few neurons like that, but not that many in this experiment at least. I'll just briefly mention further work along these lines that was done by Alden Hung, who showed that shading and disparity information like this can be used not only to infer surface shape, but to infer medial axis shape. So people in computer vision know that medial axis or skeletal or stick figure shape is very effective for representing some kinds of stimuli in our world, basically very long branching stimuli, like the shape of the human body, for example. But ultimately, medial axis shape is also derived from surface information. So it's derived from the surfaces that sort of clothe the implicit skeletal shape. So he found that there was lots of sensitivity to medial axis shape, and in some cases, almost pure sensitivity to medial axis shape. So this is an example. Again, this was an experiment based on the genetic algorithm, finding a region of shape space that a cell was sensitive to and exploring around that region. And this is just a follow-up control test for one neuron based on a high response stimulus, middle response stimulus, and a low response stimulus. And in this case, the response to this high response stimulus remained right around 60 spikes per second, regardless, or practically regardless of what the surface information that defined the medial axis shape was. So all of these responses are quite high. In fact, the highest response is to this complete stick figure shape that has almost no surface information. But what Alden found was that most cells that he studied were encoding some combination of medial axis and surface information. So here's an example here. This is the original shape from the genetic algorithm. Here are variations in which medial axis shape is kept the same, but surface structure changes, and you can see that responses go way down with the wrong kind of surface structure. And this makes sense because we need to know about both medial axis shape and surface shape, and for recognizing things like human bodies, we certainly use medial axis shape for the torso and the arms and the legs, but then the bulbousness of the head is more of a surface shape thing. So the last thing I'll talk about is work that's in revision by Siavash Vaziri. And what he did that was new was that in all these previous experiments, we've been studying basically object shape on a small screen. Objects with a size on the order of 5 to 10 degrees of visual angle and diameter. So Sia set up a projection screen, 77 degrees visual angle wide, projecting in stereo. And he created a software for generating abstract 3D shape. Well, let me first say the depth cues again were stereoscopic shading, but here also perspectival texture, which you can see here. And we realized early on that perspectival texture or texture flow, which I think some people at this meeting study, and perhaps we'll talk some about later in the meeting, really important for very large scale shape, for very distant shape, because at great distances, stereoscopic cues provide negligible information about shape, because there's very little change in disparity at great distances, and you really need to depend on something else. And we just discovered subjectively when we played with different cues that some kind of texture flows like this were critical for giving the impression of very large, even landscape-like surfaces, which we were aiming for. So his genetic algorithm, in addition to morphing shape, just like we always had, also had a high probability of morphing size. So a stimulus that looks like this could get bigger and bigger and bigger till eventually what you're looking at is a small patch of a virtually much larger shape. But when you do that, you go from what most people would call an object stimulus, that is something in near peripersonal space, subtending maybe 10 degrees of visual angle at most, topologically spherical, closed and bounded surface, to something like this, which actually extends beyond the boundaries of the viewing region, in this case the boundaries of the projection screen, and because of that is, in a topological sense, planar, unbounded, extending forever. And the genetic algorithm could range continuously across these. So if you got great response to an object, you found out if it'd be an even better or worse response if it became a landscape. But it was also constrained toward overall even sampling between the two. So it was a fair test rather than the algorithm just accidentally converging in one direction or another. So what Sia found out relates to this incredibly dense reanalysis of the organization of the ventral pathway from Kravitz, Salim, Baker, Ungerleider in Michigan. It's in ticks in 2013. It's a giant tome of a paper that collects information from over 300 other papers. They had to argue with the editors of the journal to go over the standard limit for a number of references. And so one of the most interesting things for me that comes out of this reanalysis is the separateness of channels in anterior inferotemporal cortex. So there's a channel that runs through the ventral bank of the SCS. There's a channel that runs along the lateral convexity of TE, and there's a channel on the basal surface. And these are very distinct in their connectivities with other parts of cortex, with medial temporal lobe structures, with basal ganglia. It's quite a striking story, and this paper's really changed my life, I have to say, as a researcher. So what Sia found was that while STS neurons seem to mainly be processing object shape, which is just what I would have predicted, everybody would have predicted, surprisingly, a large majority of cells, and on the lateral convexity in this middle channel here, TED, seems to mainly process environmental shape. So that's surprising because most of us, including me, have considered ventral pathway to be largely or exclusively concerned with objects, and also surprising because when people have used fMRI, to look for specific sensitivity to places, to environments, to landscapes, to buildings, to room interiors. They get posterior activation in parahippocampal areas, beginning with the definition of the posterior parahippocampal, the parahippocampal place area, the PPA, by Kanwisher and Epstein. You also get activation in human cortex and transverse occipital sulcus, and on the medial wall, retrosplenial cortex. And in the monkey, the homologs for all of this is a pathway that starts from the caudal part of inferior parietal cortex, passes through these medial wall areas, and ends up in what is probably the monkey homolog of PPA, that is TFO, TF, TH. Darius Shao at Caltech has recently recorded from areas in that region using fMRI to find the region, and then using microelectrode recording to prove that cells in this region really are very specifically sensitive to scenes rather than objects. So here's an example result from Sia's research. This is a neuron he recorded from SDS, so this is an anatomical MRI coronal slice with a tinted area indicating the region of the ventral bank of the superior temporal sulcus that he recorded from. This was the location of this particular neuron. Here are the 5 highest response stimuli from lineage 1. Here are the 5 highest response stimuli from lineage 2. They're kind of hard to see because this is really a 77-degree wide screen here, but clearly they're object stimuli, and this was very characteristic of what happened in STS. But in TED, in contrast, this was a more typical result. So here the tinted region indicates where TED was. This dot is the recording location for this particular cell. 5 highest response stimuli from lineage 1, from lineage 2, and now these are much more landscape-like stimuli that overflow the boundaries of the screen, stimuli that look like something you would walk around in or could exist in rather than stimuli that you could hold in your hand and manipulate. To give you an idea of sort of the variety of sensitivity to large-scale shape, I'll just run through, in TED, I'll run through a bunch of examples with a high response stimulus from one lineage and a high response stimulus from another lineage. Just as we have found with object shape, it is not the case that there's a single global shape that defines the response of a stimulus, but there is shared partial structure. So our initial guess, although we haven't done the analysis, is that, again, a fragment-based coding scheme would be the way that representation of environmental shape works. Some things that look like large structures, some things that look like landscapes, large structures, and some things that look like interiors. So one of the things that Sia did with his algorithm was, with some probability, it was possible to actually pass inside the shape. So object shape is largely convex. If you sum up curvature, it's going to be convex overall. The shape of this room that we're all in is if you sum up overall the curvature, it's concave. So with these very large stimuli, you could go inside them and see a shape that looks concave. And that's an example, and that's another example, and that's another example. So this is a population representation of these results. So all the cells in STS running from the fundus out towards the lip are plotted in order along here. All the cells in TED running from the lip in a medial direction are plotted here. So it's sort of like overall, as you go around here, you're going from left to right in this curve here. Every cell is represented by a colored bar. The colors themselves are just a redundant cue for stimulus size. So what's done here is to look at the average responsiveness of each of these cells to stimulus scale ranging from objects to environments. So the object range is red, the middle range is yellow, the environment range is green. In STS, cells are primarily sensitive to objects, and there are only a few cells that are responding to environment scale stimuli. In TED, just the opposite. There is some responsiveness to objects, and obviously people have studied object coding in this part of IT before, but there's a lot more sensitivity to environmental scale shape. You can do a Wilcoxon rank sum test to look at the degree to which that is statistically significant. And for a majority of TED cells, greater sensitivity to environments is significant. For a majority of STS cells, greater sensitivity to objects. These are very consistent results across lineages. So there's the correlation coefficient between two lineages for scale tuning. This is the distribution of those R values, and very much clustered toward 1 for STS and for TED. Here's all of this information collapsed into two curves. The red one for STS showing greatest sensitivity in the small object scale range. The green one for TED showing pretty low responsiveness in the object scale range, and really jumping up right at the point where stimulus diameter exceeded the bounds of the screen. So you went from a topological sphere to a topological plane. Okay, so what about the surface information that all that tuning is based on? Well, first of all, responses were very dependent on depth information. This is the same kind of plot that I showed you before for STS from Yukako's data. This is TED now, not STS. Responsiveness to environments. Here's the modulation strength with 3D cues. Here's the modulation strength without 3D cues, although with texture. So texture either flattened onto the frontal parallel plane to destroy depth, or randomized, scrambled like this to destroy depth. And even more so than in Yukako's data, you can see that while there are high values along this axis, there are not high values along this axis. So for environmental shape representation in TED, it seems like things are, if anything, more emphatically three-dimensional. Responses are not dependent on the particular type of texture. Here we halved the spatial period of the texture. Here we doubled the spatial period of the texture. But the correlation between responses under these conditions and the original conditions is quite high. Not shown here, we also changed the type of texture pattern that we put across there. Again, very robust to changes in lighting direction and resultant changes in the contrast pattern across the image, lighting from the right, from the left, from the bottom, from the top. Every dot represents a cell here, and responses for these different lighting conditions were highly correlated with the original lighting condition, which again was from the direction of the viewer. So what particular depth cues were important? Not so much stereo in this case. So if you look at, this is 22 cells we were able to do all these different combinations of depth cues on. And if you compare the response with all of the depth cues present to the response with only stereo present, responsiveness is largely destroyed in a lot of cells. And again, this makes sense in that stereo information is just less useful, less present on large depth scales. The cue that is probably strongest in this case is texture flow. So texture flow alone produces stimuli that elicit responses nearly as strongly as when all cues were present. There's some sensitivity to shading. For some cells, other cells, shading was not a sufficient cue. So the lack of stereo sensitivity in TED was actually reported in a 2000 science paper by Jansen and colleagues. And their interpretation was that this meant that TED processes only two-dimensional shape. But our interpretation would be that that conclusion is wrong because it conflates sensitivity to 3D shape with just one cue, that is stereo. Stereo is not the only cue. Our results show that, in fact, TED is very sensitive to three-dimensional shape, but on environmental scales where stereo information is not that useful, instead texture flow information is much more critical, along with shading. As I said, this is a surprising result relative to the fMRI literature, but it makes sense to me, I think, because you would want shape information about environments to be carried forward into the ventral pathway because we don't usually perceive objects in isolation. They exist in scenes, and their meaning depends on part on what scene they're in and how they relate to it. Our results also make sense in relation to the connectivity of these channels. So STSV, which is where we find lots of object information, strongly projects to ventrolateral prefrontal cortex, which is involved in object working memory, strongly projects to orbitofrontal cortex, which is involved in representing object value. And the strongest representations for TED are in the direction of the medial temporal lobe, initially into perirhinal cortex, and from thence into ultimately the hippocampus, which would make sense for spatial information. So to review, I talked about work by Yukaka Yamane showing how stereo, disparity cues, and shading cues from surfaces are used to infer 3D shape of objects, mainly in the ventral bank SDS channel. I talked about work by Alden Hung showing how the same kind of surface information can be used by neurons to implicitly infer medial axis shape. And finally, I talked about work by Siavash Vaziri showing how primarily texture flow information seems to be used to infer shape on environmental scales. And that's it. Thank you.
Thank you very much. Any questions, comments? Ishida-san.
Thank you. Excellent work. So my question is about the spatial coordinate those representations are made, and in particular for the environment one, probably the best one would be the body-centered representation. An object should be some kind of an object-centered representation. Yeah. Can you say anything
Not
about
yet. I hope we'll be able to say something about that from our analysis of these data, although it might take a dedicated experiment, which we did not do. So the reference frame for object coding based on our previous experiments is definitely object-centered and very invariant to other things. I don't have data that speaks to it, but people who do fMRI work, particularly Russell Epstein, have looked at this question of, to the extent you can look at it with fMRI, do you have more constancy for egocentric space, as you're suggesting, which would be important for navigation, or allocentric space, which would be important for recognition, being able to recognize a scene. And different regions, like retrosplenial cortex, parahippocampal place area, are more or less sensitive to egocentric versus allocentric and may have more to do with navigation versus recognition. Great question.
Thank you.
So in human perception of shape, especially from Pislow's lab, there's quite a bit of evidence that humans infer 3D shapes which are regular and symmetric in the absence of other information. Does your genetic algorithm for the 3D shapes, does it generate regular and symmetric shapes also as it goes along? Or could that be a population level stuff?
So it's possible, but the probability is quite low, except in the sense that a neuron that really wanted to see symmetric shape could potentially sculpt that in the evolving stimuli, although we've never seen that. So potentially what that means is that we should have actually introduced some bias for symmetry, and then we'd be able to answer that question better.
Yeah, well, I mean, the set of symmetric objects is a set of measure 0, essentially, when you look at all objects. So you essentially have to generate them yourself, I would suspect.
Sure, yeah, I think it's unlikely to arise by chance. Again, except in the sense that the algorithm is sensitive to what neurons are responding to, and the algorithm can do some hill climbing in any direction, including symmetry. So, I mean, it's a post hoc analysis we could conceivably do to see if there's any bias towards symmetry versus not. But I would say no. Not that we've seen, but we haven't tested it.
The final little question. So in your cells which are looking at large scale changes, you found that perspective texture was useful, but you never tried that with the smaller objects because you had very sparse informative texture.
So the smaller objects in this experiment did have texture, did have exactly the same texture.
And they didn't show up with a higher weight than the other textures?
They didn't show up with a higher rate?
You know, in the original one that you showed, texture just didn't have much weight by itself.
Yeah, although that was a different kind of texture.
Yeah, that's what I'm saying.
So this is, you're bringing up another unanswered question that we may be able to look at with this analysis, although we may not have done enough control tests. But a really interesting question. If you put perspectival texture on object, how effective, how important would that be relative to stereopsis and shading, which clearly are important, perhaps equally or more effective? I think that remains open.
Any other question? Yeah, go ahead.
Yeah, that's nice as usual. I want to ask about this unpublished part that you talked at the end. So the part if I read this right, you're saying all of what you called TED, which extends from the STS to almost the AMTS, is involved in these environmentally large stimuli and less about objects in the 10-degree space. Did I hear that right?
Yes.
Isn't that sort of, so you would predict, for instance, well, first question is, how does that map to Doris's and Simon's work that you alluded to? I didn't quite catch that. Can you first tell me that? I mean, with the MR and place areas is defined? Is that the
Sure.
same region,
Sure.
or is it?
No,
yeah.
no, no, no. It's very different. So all of the fMRI work shows, that it's dorsal pathway areas, parahippocampal cortex that receives information from dorsal pathway areas. So in talking to people that know a thing or two about fMRI, some people will tell you that there's a huge signal dropout in the temporal lobe. Maybe you know this too, due to the ear canal. So I think that's one thing that has suppressed the ability to find this area if it has a homologue in humans. The other thing that I will mention is that if you look at some of the fMRI papers, and I didn't show these figures, they show they're really big activity, and the activity they talk about in these posterior areas, but you'll see little dribs and drabs of stuff more anterior. So in hindsight, you can look at those pictures and say, I think I see some place sensitivity there.
Okay, so the MR is not clear in that case where you are, but so then my other question is that the recordings, that area that you're talking about is most of classic IT. I mean, that's the region that I'll show tomorrow. You can decode from those cells what object is in the environment. So do you think that we're just looking at the same elephant with two different sets of images and concluding different things, or how do you think there's cells interspersed, or how do you parse
so
that?
There's plenty of object sensitivity there. Are you talking about your work in particular, or?
Well, that's kind of the
as an
tip.
example, yeah, right.
But all recordings in IT are mostly from that region, much less from the dorsal bank or the ventral bank of the STS,
I'm not sure that's true.
Well, if you look at the survey of what people called IT, again, it's evolved over the years, but that's where most of, in my opinion, I mean, that's where all your previous work, I think, most of your recordings were from that region too. So
mainly from the ventral bank STS.
okay, so.
Yeah.
All right, so maybe we should talk offline.
Look, there's plenty of object sensitivity there, and so there are plenty of cells that are more sensitive to objects than environments, and even cells that are sensitive to environments also have some sensitivity to object shape. So look, I'm as surprised as you are, and initially as dubious, but we've at least tried to do every control we could, and I think it's solid. Although, again, it's not black and white.
So the prediction would be, for instance, if you lesion that part of the tissue, you would have little deficits in object discrimination tasks. That would be a prediction of this correlational inference.
So I'm not sure about that. So there's a lot of crosstalk between these three channels, despite their different connectivities to basal ganglia and everything. And I think if you lesion that part of cortex, you may be destroying an absolutely critical channel for object recognition because it's talking to STS. And I think the interesting stuff in the future for this line of research will be what's the relationship between objects and scenes? To what extent do they get put together? The stimuli that we used here were one or the other. We really want to use both and start moving, in our case, in a parameterized naturalistic way rather than using real object photographs toward things that are more like real life images and photographs that are scenes with objects populating it.
Okay, Komatsu-sensei. Just a short question.
In your earlier work with Yukaku Yamane, I thought the 3D shape is coded by the combination of surface curvature at remote locations on object surface.
Mm-hmm.
How about the coding of environment shape? Is it same?
So we haven't done the analysis, and that's why I was merely waving my hands and showing those examples. So within the next year, I'll be able to answer that question. And now all I can do is wave my hands and say that if you look at high response stimuli across lineages, you can see the same sort of agreement and fragmentary shape. But I hope that there will be lots of interesting differences in the reference frame, the biases in orientation, for example. You might predict a very strong bias towards orientation bias and gravitational coordinates. And, yeah, but sorry, I don't know yet.
Okay. With that, we are out of time, and thank you very much. Thank you.